Hello viewers, welcome to my channel. Today we will discuss about some important questions on antibody or immunoglobulin. This is one of the important topic in immunology. In my previous video, I have already discussed about some questions on this topic. For CSIR NET, ICMR NET, GATE, NEET exam, the questions related to this topic is very important. Some brief notes related to this topic is provided here, which will be beneficial for you. So stay tuned in this video. First question. Antibodies are option 1 protein, option 2 glycoprotein, option 3 carbohydrate, and option 4 is phospholipid. And the correct option is glycoprotein. Antibodies are basically glycoproteins molecule. This antibody typically have 4 polypeptide chains, that is 2 identical light chain and 2 identical heavy chain, forming the L2H2 structure. The next question. The Hinge region of antibody molecule helps in option 1 binding of antigen, option 2 binding of phagocytic cells, option 3 provide flexibility, and option 4 is none of this. Now, what is the Hinge region of antibody molecule? The gamma, delta and alpha heavy chain contain an extended peptide sequence between the CH1 and CH domain that has no homology with other domain. This region is called the Hinge region. It is rich in proline residue and in flexible, giving the IgG, IgA, IgT segmental flexibility. As a result, the two Fab arms can assume various angle to each other when the antigen is bound. That is, the Hinge region gives the IgG, IgA, IgT segmental flexibility. So the correct answer here will be the Hinge region of an antibody molecule helps in provides flexibility. Apart from the proline, another important amino acid in Hinge region is cysteine. So the two prominent amino acids in Hinge region are proline and cysteine. The large number of proline residue in the Hinge region gives it an extended polypeptide conformation, making it particularly vulnerable to cleavage by proteolytic enzymes such as papain or pepsin. For CSIR NET, ICMR NET, GATE and various other exams, very often you will get some questions related to papain or pepsin digestion of immunoglobulin molecule. I will discuss about this question later. The cysteine residues form interchain disulfide bond that hold the heavy chains together. The number of interchain disulfide bond in the Hinge region varies considerably among different classes of the antibodies. That is, the number of interchain disulfide bond varies between IgG, IgA and IgT. Even this number of interchain disulfide bond varies between the species. Here the Hinge region of an antibody molecule is shown. Next, digestion with pepsin results in option 1 2 Fab fragments and 1 Fc fragment. Option 2 Fab 2 fragment. Option 3 Fab prime fragments. Option 4 Fv fragments. Basically, pepsin is a non-specific endopeptide that is active only at acidic pH. It is irreversibly denatured at natural or alkaline pH. Pepsin cleave or digest just below the disulfide bond of the Hinge region. Digestion by the enzyme pepsin normally produces one Fab2 fragment and numerous small peptides of Fc portion. So the correct option here will be the option 2, that is Fab2 fragment. The resulting Fab2 fragment is composed of 2 disulfide connected Fab units. The Fc fragment is extensively degraded and these small peptides can be separated from the Fab2 by dialysis, gel filtration or ion exchange chromatography. The digestion with pepsin is shown here. The pepsin cleave just below the disulfide bond of the hinge region. As a result, it produces Fab2 fragment and small peptides of Fc fragments. Digestion with papain results option 1 2 Fab fragments and 1 Fc fragment. Option 2 Fab2 fragment. Option 3 Fab dash fragments and option 4 is FV fragment. Papain is a non-specific thiol endopeptidase that has sulfhydryl group in the active site, which must be in the reduced form for activity. When the IgG molecules are incubated with papain in presence of a reducing agent, one or more peptide bonds in the hinge region split. Papain cleave or digest just above the disulfide bond of the hinge region, producing 3 fragments of similar size, 2 Fab fragments and 1 Fc fragment. When the Fc fragments are of interest, papain is the enzyme of choice because it yields an intact 50,000 Dalton Fc fragment. In contrast, digestion with pepsin does not result in the intact Fc fragment. It will produce Fc fragment into small fragments. So the correct answer here will be the digestion with papain results in 2 Fab fragment and 1 Fc fragment. Here the digestion with papain diagram of this digestion with papain is shown that the papain cleave the immunoglobulin molecule just above the hinge region of the disulfide bond. It results in 3 fragments, 2 Fab fragments and 1 Fc fragment. Next question. Reduction with marcaptoethanol results in the formation of option 1 2 Fab fragments and 1 Fc fragment, option 2 Fab2 fragment, option 3 Fab prime fragments and option 4 is 2 light chain and 2 heavy chain. Immunoglobulin are made of proteins that has 2 heavy chains and 2 light chains joined by disulfide bonds, whereas marcaptoethanol reduces this disulfide bond. As a result, it produces 2 light chain and 2 heavy chain. How it cleaves this molecule and results in 2 heavy chain light chain is shown here. The disulfide bond is reduced by this marcaptoethanol and it produces 2 light chain and 2 heavy chain. Next question. Immunoglobulin which are found in asthma at elevated level. Option 1 IgE, option 2 IgM, option 3 IgG and option 4 is IgA. And the correct option is IgE. IgE is the least common serum immunoglobulin since it bounds very tightly to the Fc receptor on basophil and mast cell even before interacting with the antigen. How it involved in allergic reaction. As a consequence of its binding to basophil and mast cells, IgE is involved in allergic reaction. Binding of the allergen to the immunoglobulin E on cells results in release of various pharmacological mediators that result in the allergic symptom. Next question. Ficin generates Fab fragment by cleaving. Option 1 IgG1, option 2 immunoglobulin G2, option 3 immunoglobulin G3, option 4 immunoglobulin G4. And the correct option is IgG1, that is immunoglobulin G1. Ficin typically used to cleave mouse immunoglobulin G1 as these are very difficult to cleave with papain and pepsin. In presence of 1 to 4 millimolar or 10 to 20 millimolar cysteine, ficin generates Fab2 fragment and Fab fragments respectively. That is, in presence of 1 to 4 millimolar cysteine, ficin generates Fab2 fragments, and in presence of 10 to 20 millimolar cysteine, ficin generates Fab fragments. Immobilized ficin is a convenient reagent for producing Fab and Fab2 fragment as it avoids the need to remove the ficin enzyme after digestion. Ficin is a thyroid protease. Immunoglobulin that predominate during secondary immunoresponse is option 1 IgT, option 2 IgM, option 3 IgG, and option 4 is IgA. And the correct answer will be the IgG. That is, immunoglobulin that predominate during secondary immunoresponse is IgG, whereas the immunoglobulin that predominate during primary immunoresponse is IgM. Next, the most abundant antibody found in serum is option 1 immunoglobulin A1, option 2 immunoglobulin G1, option 2 immunoglobulin G2, and option 4 is immunoglobulin G3. And the correct option is immunoglobulin G1. Among the 5 different subclasses classes of isotypes of immunoglobulin, that is immunoglobulin G, immunoglobulin A, immunoglobulin E, immunoglobulin D, the immunoglobulin G is the major most abundant antibody found in the serum. Thus, immunoglobulin G also is divided into 4 different subclasses: IgG1, IgG2, IgG3, and IgG4. Among this immunoglobulin G, the immunoglobulin G1 is the most abundant antibody found in the serum. That is, immunoglobulin G is divided into 4 different subclasses. That is 4G, 4G1, G2, G3, G4. How can you remember that immunoglobulin G has 4 different subclasses? You just remember the 4G network. So that is 4G, immunoglobulin G has 4, G1, G2, G3, G4. Whereas immunoglobulin A has 2 subclasses, A1 and A2. Other immunoglobulin, that is M, D, E, does not have any subclasses. Next, which is the largest immunoglobulin in size? Option 1 IgA, option 2 IgG, option 3 IgM, and option 4 is IgD. And the correct option is IgM, because IgM is sometimes found in a pentameric form. IgM is the largest antibody with 10 paratopes. If the question comes that which antibody has highest number of valency, the answer will be M, because it is a pentameric structure. Even if the question comes which immunoglobulin has highest number of paratope, same answer. It will be the immunoglobulin M. Whereas immunoglobulin G are smallest antibody, but most common and most abundant antibodies of all the antibodies in the body. Next, the least abundant antibody found in serum is option 1 immunoglobulin A, option 2 immunoglobulin G, option 3 immunoglobulin E, option 4 immunoglobulin D. And the correct option is immunoglobulin E. Immunoglobulin E is responsible for the allergic reaction. It does not activate the complement pathway. It is the monomeric antibody that accounts for only 0.002% of the total serum. Next question. Immunoglobulin is associated with anaphylactic delayed type hypersensitivity reaction is option 1 IgD, option 2 IgM, option 3 IgE, and option 4 is IgA. And the correct answer is IgE, that is immunoglobulin E. It is immunoglobulin is very important in asthma, allergic reaction, atopic reaction, type 1 hypersensitivity reaction. Next question. What is the similarity between IgM and IgG? Option 1 complement fixation, option 2 placental transport, Option 3, heat stability at 56 degrees centigrade. Option 4 is sedimentation coefficient. As you know that both IgM and IgG antibodies fix complement, but IgM is the better in fixation of complement than IgG. So both IgG and IgM antibodies fix complement. This is a similarity between these two antibodies. So the correct option is in complement fixation. The similarity between IgM and IgG is complement fixation. In contrast, the IgA fraction did not bind the complement. Only the immunoglobulin G can cross the placenta. At the option placental transport is not correct because only the immunoglobulin G cross the placenta. So if the question comes, the immunoglobulin that can cross the placenta, the answer will be immunoglobulin G. IgM is the largest antibody and immunoglobulin G is the smallest antibody. That is, the size of IgM and IgG is different. One is largest antibody, that is IgM, and one is the smallest antibody, that is IgG. Thank you. Please like, share and subscribe to this channel.